Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine this. A medication you might have heard of for, well, a really common childhood issue. Something easily treated.
Okay.
Now, picture that same kind of drug showing real, unexpected potential against a very aggressive, quite deadly form of skin cancer.
Wow. Okay.
Merkel Cell Carcinoma. It's a cancer where, honestly, the current treatments often just don't cut it. Sounds surprising.
It does sound surprising. A childhood ailment drug for aggressive cancer.
Exactly. And that's precisely what we're diving into today.
So, Merkel Cell Carcinoma. Maybe we should start there. It's not melanoma, which most people know, but it's serious.
Yeah, definitely less common, but as you said, very concerning.
It is. It's a neuroendocrine cancer, meaning it starts in hormone-producing cells in the skin. And the scary part is how fast it grows and spreads.
Faster than melanoma, even.
Often, yes, which contributes to higher mortality rates, unfortunately. And the standard treatments we have, surgery, radiation, even some of the newer immunotherapies, they work for some, but not consistently enough.
So there's a real gap, a need for something new.
A critical need. Which brings us, I imagine, to this new research.
It does. We're looking at a health news report from the University of Arizona and also a pretty detailed scientific paper from the Journal of Clinical Investigation.
Okay, solid sources.
They are, both highlighting some really cutting-edge work from the University of Arizona Cancer Center. Our goal today really is to unpack this research, try and understand why this long-standing drug for pinworm infections is suddenly looking so promising against this tough cancer.
Right, and that drug is called Pyrvinium Pamoate. Like you said, Food and Drug Administration approved way back in 1955 for pinworms.
1955, so it's been around a long time.
A very long time. But what piqued researchers' interest wasn't just its age, it was that people had already noticed some, let's say, anti-tumor activity in other cancers before.
Oh, really? Which one?
Things like breast, colorectal, pancreatic, even bladder cancer. So there were hints it might do more than just treat parasites.
Okay, but still, the big question is, how? How does a drug designed to fight a parasite possibly work against cancer cells? What's the thinking there?
Yeah, that's the crucial question. The researchers had this hypothesis, this idea, that maybe some antiparasitic drugs could work against cancer because, well, tumors and parasites sort of face similar problems inside the body.
Similar problems? Like what?
Both need to hijack the host's resources, right? Nutrients, energy, space. They need all that to grow and multiply.
Okay, I see.
So the thinking is, if they happen to use similar molecular pathways, similar internal machinery to get those resources,
then a drug that messes up that machinery in a parasite
might just mess it up in a tumor cell too.
Yeah.
It's like finding the same key opens two different, but maybe similarly designed locks.
That actually makes a lot of sense when you put it that way. So, okay, how do they link this specific drug, Pyrvinium Pamoate, to this specific cancer, Merkel Cell Carcinoma?
Good question. They didn't just pick it out of a hat. The researchers had previously figured out that a specific cell communication system called the Wnt signaling pathway, that pathway is really important in tuning normal skin cells into Merkel cell carcinoma.
Wnt signaling, okay.
And it turns out Pyrvinium Pamoate was already known to interfere with that Wnt pathway.
So that's the connection.
That was the light bulb moment, yeah. It was logical to then ask, if it messes with Wnt signaling, could it also stop Merkel cell carcinoma from growing or, acting like cancer? Think of the Wnt pathway like a set of instructions for cell growth. In this cancer, those instructions are scrambled.
And Pyrvinium might unscramble them, or at least shut them down.
Something like that. Yeah, disrupt those faulty instructions.
So what happened when they tested it? In the lab first, I assume.
Exactly. In lab models using Merkel cell carcinoma cells, they found Pyrvinium Pamoate did inhibit growth. It slowed the cancer cells down.
Okay, that's promising.
And maybe even more interesting, it seemed to actually reverse some of the specific features that make these cells neuroendocrine cancer cells.
Reverse them, like pushing them back towards normal.
Sort of, yeah, it was quite striking. So with those encouraging results in the dish, they moved to living systems, mouse models.
Right, the next logical step. What did they find in the mice?
Also promising. In mice, they've had Merkel cell carcinoma tumors. Giving them Pyrvinium Pamoate actually reduced the tumor growth. The tumors got smaller or grew much slower.
Okay, so it works in cells, and it seems to work in a living animal model, too.
Correct. Which brings us back to that Journal of Clinical Investigation paper you mentioned. They really dug deep into the how at the molecular level there.
Right, using some advanced techniques.
Absolutely. They use something called ribonucleic acid sequencing analysis. Basically, it lets you see all the genes that are active or switched on in a cell at a specific time.
Okay.
They did this on normal human skin cells, fibroblasts, that they'd engineered to make proteins from the Merkel cell polyomavirus.
That's the virus linked to a lot of these cancers, right?
Exactly. And this sequencing showed specific changes happening in that WNT signaling pathway because of the virus proteins. Then they used powerful computer analysis bioinformatics to sift through all that data. And the analysis pointed straight at Pyrvinium Pamoate as a drug that could potentially reverse the specific gene activity pattern seen in Merkel Cell Carcinoma.
Wow. So the computer analysis basically flagged it as a potential fix.
In a way, yes. Like molecular detective work, identifying the cancer's fingerprint, and then finding a suspect Pyrvinium that could smug or change that fingerprint.
Okay, this is where it gets really fascinating, because it sounds like it wasn't just about the WNT pathway. Pyrvinium was doing other things too.
That's right. It turned out to be a multi-pronged attack, which is often what you want in cancer treatment.
Makes it harder for the cancer to escape, I guess.
Precisely. So besides modulating WNT signaling both the main pathway and an alternative one, it also hit the cancer cells by triggering programmed cell death, which involves a famous protein called protein 53.
Ah, the guardian of the genome.
That's the one. Also disrupting the mitochondria, the cell's power plants, and causing stress in another part of the cell, the endoplasmic reticulum, which is crucial for making proteins correctly.
Okay, let's unpack those a bit. This protein 53 pathway activating that sounds like a good thing against cancer.
It generally is. Protein 53's job is to stop damaged cells from becoming cancerous, often by telling them to self-destruct apoptosis. Cancer cells often find ways around this.
Right.
Pyrvinium seemed to boost protein 53 levels and trigger this self-destruct sequence in Merkel cell carcinoma cells that had functional protein 53.
But what about cells where protein 53 is already broken, which happens in some cancers?
That's a key point. Interestingly, Pyrvinium also showed cell-killing effects in cell lines lacking functional protein 53.
Oh, so it can work either way.
It suggests it can, yes. It implies there are protein 53 dependent and independent ways it triggers cell death, which is a big plus, potentially making it effective even if that specific guardian pathway is down.
Okay, that's significant. And the mitochondria, you said it disrupts the power plants.
Yeah. Cancer cells are energy hogs. They grow so fast, they need loads of energy from their mitochondria.
Makes sense.
Pyrvinium was found to significantly reduce the mitochondria's ability to produce energy through cellular respiration. It also lowered the levels of key protein components within the mitochondria.
So it's like cutting the power lines to the cancer cell factory.
Pretty much, starving them of energy.
And the last one, endoplasmic reticulum stress. What does stressing that part of the cell do?
The endoplasmic reticulum, or ER, is like the cell's protein folding factory and quality control center. If it gets stressed, say by too many misfolded proteins, it triggers the response.
To try and fix the problem.
Initially, yes. It's called the unfolded protein response. But if the stress is too much or goes on too long, it flips a switch and tells the cell to die instead.
Ah, another route to apoptosis.
Exactly. Pyrvinium increased the markers of this ER stress and seemed to mess up that unfolded protein response pathway too.
Pushing the cells towards death again.
It seems so. And interestingly, the researchers noted that Pyrvinium actually caused a stronger ER stress response in these cells than other known ER stress-inducing chemicals they tested.
Wow. So it's hitting the WNT pathway, triggering cell death via protein 53 and maybe other routes, cutting off energy supply and causing massive stress in the protein factory.
It really seems to be attacking from multiple angles.
That sounds incredibly promising. So stepping back, what are the big picture implications here?
Well, the most obvious one is repurposing, right? Taking an old Food and Drug Administration approved drug like Pyrvinium Pamoate with a known safety profile for its original use.
And potentially using it for a really tough cancer.
Exactly. That could mean a much faster path to clinical trials and potentially helping patients sooner than developing a brand new drug from scratch.
Which can take years and billions of dollars.
Absolutely. And the fact that it works through multiple mechanisms like we just discussed is also a huge plus.
Why is that so important?
Multi-target drugs can often be more effective, just hitting the cancer harder. And critically, it might make it much harder for the cancer cells to develop resistance. If you block one road, they might find a detour. If you block several roads simultaneously.
It's much harder to get through. Makes sense.
And then there's that protein 53 point we mentioned.
Right, that it works even if protein 53 isn't functional.
Yes. While mutations in the TP53 gene, that makes protein 53, aren't as common in Merkel cell carcinoma as in some other cancers, they do happen. So having a drug that isn't completely reliant on functional protein 53 could broaden the number of patients who might benefit.
So for people listening, why should they care about all these, intricate molecular details, what pathways, mitochondria?
I think understanding this stuff gives you a real appreciation for, well, first, how complex cancer is, but also how incredibly clever researchers are being in finding new ways to fight it.
Yeah.
It shows the power of looking at fundamental biology and finding these unexpected connections. Like, who would have thought a pinworm drug could teach us so much about fighting skin cancer? It highlights the value of basic science.
Absolutely. But as with all early research, especially this promising, there have to be limitations, right? Things we still need to figure out.
Oh, definitely. And the researchers are upfront about that. They stress that more work is needed to figure out the best way to use Pyrvinium Pamoate specifically for Merkel cell carcinoma. Dosing, timing, maybe combinations, all that needs further study.
And what about the models they used? Any limitations there?
Well, the initial cell work used fibroblasts, which might not be the exact cell type where Merkel cell carcinoma sprang.
Right.
That's always a consideration.
But they checked it in other ways, too.
They did. They confirmed key findings in neonatal human dermal fibroblasts, which are thought to be a more likely cell of origin, and also by analyzing data from actual human Merkel cell carcinoma tumors, which adds confidence.
Okay. And the mouse studies. Any issues there?
There was a note about administration. When they injected higher doses into the abdominal cavity intraperitoneal injection, the mice didn't tolerate it perfectly.
Ah, so a potential issue for giving it to people.
Potentially, with that method. However, they also pointed out that other studies, maybe for different conditions, have shown that giving Pyrvinium orally seems to be safer and better tolerated.
So that might be the way forward.
It could be. That needs optimization too, but it suggests the tolerability issue might be solvable with a different delivery route.
It's always good to hear directly from the scientists. Did the lead researchers offer any key takeaways?
Yes, Dr. Megapati, the lead author, really emphasized that Merkel cell carcinoma incidence is rising, and because it shares traits with other aggressive cancers, finding new treatments is just vital.
And did she comment on the parasite connection?
She did. She mentioned that interesting parasite-like idea, that shared need for resources, as a potential underlying reason why an antiparasitic might work against cancer. It's a fascinating hypothesis.
And the overall conclusion from the Journal of Clinical Investigation paper.
The authors basically concluded that their work really deepens our understanding of how WNT signaling drives this cancer, and importantly, it provides strong preclinical evidence that Pyrvinium Pamoate has real therapeutic potential for Merkel cell carcinoma.
Okay. So as we wrap up this deep dive, what's the final thought we should leave our listeners with?
I think it's just the sheer possibility here. An old, familiar drug, used safely for decades, for one thing, now potentially having this power against a deadly cancer by hitting it in multiple ways.
It makes you wonder what else is hiding in plain sight in our medicine cabinets.
Exactly. What other existing drugs might have unexpected talents against complex diseases like cancer or Alzheimer's or others? Thinking about these fundamental biological pathways, these shared mechanisms, could unlock so many new treatment strategies.
It's a really exciting prospect. And if you listening want to dig deeper into this specific research, definitely check out the paper in the Journal of Clinical Investigation. The digital object identifier is 0.1172/JCI177724.
And you can also find more details in that health news article from the University of Arizona.
Absolutely. Keep exploring, keep learning. It's a fascinating time for this kind of research.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.